All right, good afternoon, everybody. My name is Russell Bodily. I am an incoming D1 dental student, actually, and I had the wonderful opportunity to work with Dr. Creamer and Dr. Staudinger this summer, along with all my other colleagues at the MSSU KCU Research Consortium as an NIH R15 grant fellow. I focused on the emerging roles of the pregnetin X receptor expression and activation in triple-negative breast cancer cells. So first off, I wanted to focus on explaining what the pregnetin X receptor is. So as was described earlier a little bit by student Dr. Whitney, the pregnetin X receptor is extremely important in the liver and the colon. It's an important nuclear receptor in both of these areas and some other tissues as well. It's a ligand-activated member of the adopted orphan nuclear receptor superfamily, and it plays a large role in activating proteins important in the xenobiotic response, such as oxidative metabolizers, conjugation, and transport proteins. I want to take a second to focus on some of the activators for this nuclear receptor. Rifampicin and St. John's wort, which was talked about a little bit earlier. These are known agonists for PXR, and I'll talk a little bit about why that's important in a second. So what we did was we looked into some of the literature about this PXR in regards to breast cancer. And in 2009, there was a paper that suggested PXR is expressed in human breast cancer. And they showed this using Western blots and immunohistochemistry. And they also were able to show that some of the downstream targets for PXR were also upregulated when activated by something like rifampicin or St. John's wort. In 2021, another paper suggested that when PXR expression is high in breast cancer patients, their prognosis is much lower. And so this led us to ask the question, why is this happening? Because none of this research was showing why this was happening. Also, another thing that they showed was not only in triple-negative breast cancer, but also a couple of other cell lines, PXR was also expressed, which you can see right here. So we looked into it a little bit further using an NIH gene expression omnibus database. We found that both in normal tissue and tumorous biopsies of breast patients, PXR expression was there. We could see that through these counts. And so with our funding, we got a hold of a triple-negative breast cancer cell line that was reported in one of those other papers, and we tested it. And we saw right here, this is the MDA MB231, this is the triple-negative breast cancer cell line, and it had an extremely low expression of PXR, even though the papers were saying that it had a high expression. So with the ones that we got, we just had a low expression. So to make it a good model, we had to introduce a stable overexpression of this PXR gene. And so we did that by using a zeaxanthin selective vector. We engineered a stable and selective human PXR cDNA to transfect into our breast cancer cells. And we made 3 separate clones. And with those, once we had those clones, we wanted to make sure that PXR was actually there. And so we validated this through real-time quantitative PCR and our Western blotting. And we were able to see that there truly was PXR present, and where wild type there was none present, as you can see in these 2 figures. We also validated it. We have validated this information with immunocytochemistry as well. The fluorescence depicts that the presence of PXR is much more vibrant than in the wild type MDA MB231 cells. And so we started off by testing, okay, we wanted to find this pathway, the molecular pathway that was causing this poor prognosis. And so we started with the canonical PXR genes, these downstream targets, and we saw a very low, we found this wasn't the reason. There was some overexpression when PXR was activated. However, this isn't the story that we found. And so we decided to get a broader scope And by doing that, we took wild type vehicle and rifampicin treated cells, and then also the overexpressed vehicle and rifampicin treated cells. And we sequenced them using Illumina RNA sequencing and ran statistical analysis. So these next couple slides are going to talk about that statistical analysis. We ran about 6 statistical analyses. The first was our principal component analysis plot. And in this, we see the wild type cells, both rifampicin and vehicle treated, all clustered together. Now this scale, it shows a variance. And when PXR was overexpressed, we saw that they varied extremely from the differentially expressed genes from the wild type. We also saw that the rifampicin treated cells differed from those that were just vehicle treated. So we know that there is definitely something different going on with the PXR activation. And so some more proof of that is this volcano plot was showing the wild type rifampicin versus vehicle, and then rifampicin versus vehicle in the PXR overexpression. These are all differentially expressed genes. We saw that there was both downregulated and upregulated genes. And so we wanted to focus on that and say, all right, what is actually happening? So we went to differentially expressed gene heat map. This was really cool to see that in our analysis, it separated everything for us, very much what we were hoping for. PXR, when activated by rifampicin, differed extremely from everything else. The overexpressed PXR, when treated with vehicle, was much more similar to the wild type. And so we found a couple of the top hits, which I'll talk about a little bit later, and validated those as well. So we went into, once we had all this information, we knew PXR is 100% causing something or changing something with this breast cancer. So we did a KEGG analysis, a KEGG pathway enrichment analysis. This gave us a couple of overall pathways that we could focus on with the differences from those genes that we were targeting. And then to dive in a little bit deeper, these ones that were highlighted came up a lot more with this gene ontology. There were 3 figures for this, and in all 3, with the gene ontology biological processes, the cellular component, and the molecular function, we were able to make a figure from this. And we found that PXR is driving cell migration signaling. It's causing a lot of upregulation in these genes that cause this migration. And so what this could mean clinically is, if this is actually happening, the poor prognosis may be becoming because of metastasis, a higher amount of metastasis that is caused by PXR activation or expression. So with those genes of interest, the ones that were some of those top hits, all of them had to do with motility in some way, or cytoskeleton changes. So the log fold changes that were given to us by RNA sequencing, we wanted to validate those and see, is this actually what's happening, which we did via real-time qPCR. And everything that was activated by rifampicin had an upregulation, just like we were seeing in that RNA sequencing. So some of the future things that we're going to do, we're actually starting some matrigel invasions and scratch assays to test this motility. Is this actually what's going on? And then we're also going to test some small or siRNA targeting to see if the decreases in rifampicin induce migration. And so this research wouldn't have been possible without all of these people, so I wanted to give them a second of thanks as well.
So on that heat map you showed, I saw one that was at IGF as well. It looked really low in the wild types and really high in the mutated ones.
Which one was it?
IGF-N1, looks like the 4th or 5th one down on the top.
Oh,
from the top. Sorry.
Yes, right there.
Yeah. So you talked a lot about the idea of motility. Did you think of, I mean, especially with this being triple negative cancers, is there any link with IGF-1 overexpression and things like that you saw?
I didn't focus on IGF N1 specifically. Dr. Kramer might be able to answer that question a little bit better. I'm sorry.
So, so when you look at a heat map, yes, you can pick out individual genes that maybe look interesting, or we see differential expression. The way we've moved forward with what student Dr. Bodley presented here was really taking all differentially expressed genes that hit our log two-fold, putting that through KEGG and GO. This is a representation of the top 30 differential. This doesn't show all of those migration and motility genes. So we do have plans. I've got about 15 different folders with all the different analyses that we're in comparisons that we're starting to do from one data set, which is the power of RNA-seq, right? You get terabytes of data. You just have to have the time to then follow up. We're starting on the ground floor with the most obvious thing that came from GO and KEGG, which was migration.